So thank you so much, Martin, for your invitation to this exciting carbohydrate days, 2 days. So I'd like to open this work by telling you about a story that started pretty much last year at the very beginning of the pandemic, at least when we realized that this was a very difficult, it's going to be a very difficult time. So we promptly started our simulation as soon as enough data were available. And that the first part of that simulation, which I'm going to touch upon in this talk, is what I like to call episode 1, Beyond Shielding, where glycans we found made a functional contribution to the opening of the spike. So I'd rather this time actually discuss more about our ongoing work that is available on bioRxiv, about how the glycan shield topology and the type of glycan shield actually is able to fine-tune this function of the spike. So fine-tuning the activity, the opening stability of this spike, therefore the infectivity of the virus ultimately. So my talk today is organized in this around this point. So I'm gonna talk about a bit of the COV2 spike architecture. I'm gonna mention in particular three glycosylation sites, which are, we discovered extremely functional. So I'm gonna start my video. So, which are 234, 165, and 343. So you don't really need to remember those numbers. I'm going to mention several times around, so like you probably end up remembering them anyway. So, and especially at that site, what we asked ourselves is that once they're recognized as functional by simulation and experiment, are all glycoforms equally functional? So all these are different type of glycans in those positions do confer the same function or not. So, and then we looked at some changes in topology because the virus evolves and everybody talks about the evolution of the protein, forgetting that the evolution of the protein involves the evolution of the glycan shield itself by mutation of glycan sites, glycosylation sites, and so forth. So we looked at that, and in particular, one loss of glycosylation acquired recently in COV2 being the first spike that has that, that carries that mutation. And then based on this, I'm gonna make some conclusions and give you some perspectives. So first and foremost, spikes. So a spike protein, as everybody knows, it's called spike because it stems out of the viral envelope, like protruding from the membrane. And we chose like this blue color to color like the spike as a blue color to highlight the density of the glycan shield. Here is a close-up image of the atomistic model of the whole spike, where in blue represented the glycans that you can see cover pretty much the entirety of the spike protein. And you can see the spike protein here if I am allowed to shave the glycan shield for a second. So as a glycoscientist, I don't like this, but I'm going to use it for one slide just to highlight some intriguing points that triggered our curiosity pretty much right away last year. So the spike protein can be topologically divided in 2 subdomains, S1 and S2. That you can see on the left-hand side, and this kind of mushroom fold is produced or obtained by the intertwined interlocking of 3 protomers, identical protomers. So the activity of the spike is actually hinging on the exposure of one receptor binding domain that you can see here in cyan, highlighted in cyan. And what intrigued us immediately when we saw this cryo-EM structure was the actual enormous void that is created by the opening of this RBD. So as the RBD lifts up, so to bind, like to adopt functional or let's say functional like structure that can bind the receptor, the ACE2 receptor, it leaves a massive void within the core of the spike trimer. And this void is so large that it reaches the very deep like core of the trimer. So in it, like from an architectural point of view, like weakening the whole structure or stability of the spike, especially in view of the fact that in COF2, the furin cleavage site comes already cleaved. Therefore, the S1 and S2 topological domain are separated already. So what we thought immediately, that was around like February, March of last year, is that the presence of the glycan shield can not only have an immunological defense role, but it also could stabilize the open spike, in particularly kind of compounding it, like functioning a bit like putty. So filling those holes and stabilizing the overall architecture. So what we did to do, to study that and to check if that was actually the case, well, we had to reconstruct the glycan shield from scratch because, as all of you know, structural information on the glycans are not available, or not like generally available. They're rarely available, and definitely not on viral shields, unless the glycans are highly restrained in terms of their dynamics. So what we did, we identified the N-glycosylation sites and O-glycosylation sites available here on the sequence of one of the protomers I highlighted in blue, the N-glycosylation sequence. Here you can see there are 22 per protomer, and from early and later, like glycosylation profiles, this one is the one that we used throughout, that is from Max Crispin's lab. All of them are like 18 at least of 22 are occupied with different glycans, mostly but with complex N glycans. And based on this profile, we reconstructed the glycans. In particular, what we did is just like for our simulations, we used our library that is under construction for like to open access availability to all of you, that contains all pre-equilibrated glycan structure that we structurally aligned on existing GluCNAC residues on the cryo-M structure. And so forth, we produced a completely glycosylated spike, which we proceeded to simulate. So how can these atomistic simulations help? So what kind of questions can we answer for you? Like viewing, like being able to reconstruct it, to view the entirety and the complexity of the glycan shield is pretty much what we can do is definitely define what is the protein accessible surface area. And we explore this quite at length in our first episode 1 work, where we analyzed what is the accessible area to antibodies or to smaller molecules that is like not covered by the glycan shield when the spike is open or closed. So when it's open, it's actually the RBD or receptor binding domain is what is the part of the spike that is mostly accessible, and it actually is a part of the essential part of the design for the existing vaccines nowadays. So this is important for finding drug accessible sites or druggable sites and for recognition of epitope from antibodies. So aside from this, what was unique about or revealing about our simulation is the role, the functional role of the glycan shield, which is like pretty much unknown previous to our research. So like the glycan shield has not only an immune defense role, but it also props up the open RBD. And in particular, this propping up of the open RBD is the work of 3 sites, N-glycosylation sites in particular. So it's the N165, N234, and N343, which are color-coded here, just to differentiate them from one another, and that they interact differently with the glycan shield, kind of like propping it up. So the question, like this particular work was analyzed at length in the first paper that we put out like last September in ACS Essential Science. And the second questions that we had, and it can be addressed by simulations, is the following. At these sites are all type of different glycans. As you know, there are multiple types, very many types, are all equally functional. Or the glycosylation type affects the ability of this of the spike to work, which is actually a fundamental question, and I'll explain why. So what we went on to try to understand is like why those glycans were there in the first place, like according to their profile given by Max Crispin here. And this particular glycosylation site was one of the most kind of like, together with the they say large vacuum that it was open, left open by the lifting up of the RBD within the protein architecture. The presence of a high manos, large I manos such as MAN9, MAN8, and MAN7 at position 234 was actually one of the most intriguing points from the very beginning, from the first time probably that I've read that Max Crispin's paper. As you can see, it's very unique. So it has a very peculiar type of glycosylation. So the first question is like, why is that MAN9 or MAN8 there? So the simulations can answer that question. So if you see here, actually, like this is a quick like movie of the dynamics where the MAN9 at all the three positions, remember this is a three protomers, identical protomers. The ones that are closed are shown in purple. Like, so the MAN9 from the RBD enclosed sites are purple. You can see that during the dynamics, these glycans kind of occupy like a cleft, like so on the surface of the protein, and they're really not accessible. They do not protrude towards or through the solvent. Therefore, they remain kind of like immature because of their difficulties from glycotransferases and hydrolases, getting them and functionalize them. And this kind of gets even worse if the RBD is open. So this is a simulation of the open side of the RBD. The open RBD is in orange, and you can see the purple MAN9 that actually crawls in, literally, that's a word that we like to use, because it's actually a crawling of this glycan through the wall of the cleft left open by the lifting up of the RBD, and it fills its entirety. So as you can imagine, you obviously no chance of any enzyme getting to it when it's inside. And it fills the hole, as I mentioned in the probably one of the first slides, so well that the RBD is in open conformation and is stable in open conformation. So the simulation that I'm kind of outlining here, the results on the left, is it is one of the ones that actually showed a very quick crawl. Like this MAN9 flies in quite quickly, like for the time scales that we're used to. This is around 250 nanosecond. This is like the crawl has ended, like with the MAN9. Interacting with the core residues of the helices at the center of the trimer, and this is facilitated by the terminal monosaccharides at the top of the MAN9. Meanwhile, the initial interaction that triggers this crawl is actually triggered by core saccharides, like in the 1,6R of the MAN9. So what we, like in the first paper, what we thought after we observed this, that, can happen in 250 nanoseconds, can also happen in 3 microseconds we've seen, so it can be fast, it can be very slow in simulation time, that we saw that position and that large oligomanos at that position was essential for stability. So what we did, we mutated that in the simulation, and what we observed is that the mutant that had just, well, in this case it is a double mutant, but even a single mutant had corresponded to a very unstable RBD compared to the you know, natural or native glycosylation, where the RBD adopted conformation, they were absolutely not the same as the normal, or like the wild type, let's say, closed and open spikes. So what we went through, it was to contact Jason McLennan, and what he did in his lab was to do this mutation and to measure binding of ACE2 by biolayer interferometry. And what he obtained is that the mutant, in particular 2N234, the one that has the MAN9 mutant, so let's remove that glycosylation. Just the only difference was that just that one site was non-glycosylated, reduced binding by 60%. So this was pretty much a huge flag up in saying, like, improving that the glycan shield is indeed functional in the working of the spike protein, in the infectivity of the virus. So the question 3 at this point is like, are all glycoforms equally functional? So what that, it was a MAN9, which is like together with MAN8, and you can see as all terminal arms, like arms with monosaccharides all occupied. What if I use like, or what if we put a different glycan there, like smaller glycan, for example? So that would would that change the function of the spike? Well, we think so. Like, so not all glycans are equally functional, and just because of what I said earlier, that let's say the filling, complete filling of the pocket hinges on a large oligomannose being there, one that can that would extend enough so that the terminal monosaccharides can interact with that helix fold that is at the core of the spike. So if I put a MAN5 or a MAN3, it just won't reach. It's just too small, and it would not reach. Like, it might just hang out at the gate, and that's but the core of that hole is not filled in the case of this type of glycosylation. Also, one thing that we should always remember is that these glycans are not 2D pictures. They do not extend like vertically, like as in a 2D SNFG kind of image. Let's not be fooled about it. Actually, they have a very different 3D structure, where the MAN9 is very much like a tree, like a bushy tree, and the MAN5, the arms are are not interacting with each other. And therefore, this means that this interaction that the glycan makes with the proteins are very different. Like, the pattern is very different. Like, aside from the size of it, so the contact maps that this glycans can make with the proteins are very different when you change them, for example, from MAN5 to MAN9, to MAN5 to MAN3. So this is not just an academic question. This is quite important, because there is a variety that has been actually reported. This is in this case is for like, it's a recent work that has been published in ACS Central Science, actually at the same time, on the Oxford AstraZeneca vaccine, and on the virus itself, and on recombinant proteins, that shows that there is a different glycosylation in different types of spikes, and that depends on the accessibility of the protein, as you can imagine, by the glycotransferases and glycohydrolases, actually making the MAN5 the most probable glycosylation site at N234 for the viral protein and the vaccine protein, in this case for the AstraZeneca. So let's put that MAN5, sorry, on, and see what happens. So as we predicted, like the simulation that here count for over 5.5 microseconds, this is just production that is counted. And here compared against the MAN9 shows a higher instability or higher dynamics rather of the spike. RBD that is mobile, like so, like to note, this trimer folds quite tightly. So this variation, the angle variation, are subtle, like in terms of this axial angle and lateral angle that we chose as coordinates to analyze these MDs. So what happens is that the MAN5 is only able to reach the gate of the cleft when the RBD is open, not the entire cleft because it's too small. It's kind of in some simulation it partially closes, but it definitely way more dynamics. But it's still competent. It really, like if you see like the comparison with the MAN9 very stable spike, you can see that it's still like in an open conformation. And this is because of the effect of these two glycans that are N343 and then 165 that can keep it open in a kind of an intermediate state, not super open or very open as you can see in the cryo-EM, but kind of intermediate. So it's still competent. So functionally, like to summarize, the smaller MAN5 cannot occupy that cleft, so like allows a higher dynamics of the RBD, which is still competent in exposing it, but not as competent as a MAN9 would be. So, and we proved that but the role of this other two glycans, there are 165 and 343, by removing them, rather removing them, changing their glycosylation from like a complex as it is normally to a MAN5, so making kind of like all MAN5 spike. And this is extremely unstable. You can see like we can explore within 5.3 microseconds in different replicates. We can explore a much higher dynamics of the RBD that is really extends over to the other side, like or to the, let's say, back of the screen. And you can see here like how big this vacuum or this cleft becomes and how unstable it is and how smaller this glycans, and unfortunately unable to prop this up. So, and this instability is actually in agreement with recent data from Robert de Vries lab that produced an immaturely glycosylated variant from GNT1 mutant cells. So this is all good. So what if we get to even smaller? MAN3 is able to just interact with the outside of the cleft. You cannot get in, and it actually leaves that cleft completely empty. And what we see in some states is actually a closing, like so almost closing, which is actually incredible for to observe with let's say conventional MD, so not enhanced sampling. And here you see the gray would be the drift that we see in the RBD within 4.3 microseconds in different simulations relative to the cryo-EM structure where the RBD is propped up. So it's quite a large conformational change for this time scale and for an MD that is not biased in any way. So the MAN3 would be completely unstable compared to the HEC like S2P glycoform, which is the MAN9. So what about, like, this is the last part, like what about the changes in topology? Well, as you can see in the video here on the left, the protein evolution of the viral protein evolution encompassed a lot of changes in the glycan shield throughout that. Is getting way more complex in our view, at least, like we're looking at the evolution site. So one aspect that we looked at in particular is the loss of glycosylation at position N370, which is the main difference or let's say like the difference that is the most functional, or that has like, you know, most functional properties from SARS-CoV, which is like the 2003 variant, and CoV2 that is now. So the RBD in CoV2 has 2 glycosylation sites, in CoV1 or CoV as 3. And N370 is one of them. So this is the N370 position, and I put it relative to the position of 234 for you to understand. Also, N370 is conserved as a glycosylation site in currently circulating or existing types of SARS-CoV, which is the pangolin version, and the RATG13, that is the bat. And you can see here that only CoV2 has this mutation that is like T372A, that caused the loss of glycosylation at that position. So what we did is like we used the most stable of our conformers, like which is like, well, not only of our conformers, but the most stable spike that you can have, that is like the HEC2S2P variant, and we put in an N370, in particular complex type, as it is found also by my script-based analysis in CoV1. And what happens is like from the side where the N370 is open, we find that the N370 actually is able to fill in like that cleft. So it actually, because of its position, is able to fit in the cleft and stabilize extremely effectively the RBD up. And this is probably one of the reasons why it's been conserved for so long, until the position 234 came around and kind of fulfills that its role, or that role coming from another side of the protomer. So what happens in the other sides? So like it's extremely stable, like it just actually, glycosylation in 370 is extremely advantageous because it props up the RBD. So what about the other sides, the one that are closed? So if you look at the closed promers, you see the RBD here, the RBDC in particular in this video that is highlighted in white. Like you can see the N370 that it fills a cleft like on the surface of the RBD, and this has been analyzed for multi-microseconds, multiple replicates, and you see that that glycan at N370 is perfectly housed. So the 1,6 arm is perfectly housed in this cleft. There is what we like to call a quintessential glycan binding site. It has like a number of hydrophobic and hydrophilic contacts that actually line up that that complex arm, the 1,6 arm, and these features that make this glycan binding site so effective are conserved throughout all these different types of COVID, species, well, strains rather. So what is intriguing is that this glycan, the N370 that binds the cleft, actually comes from another RBD. So it pretty much functions as a lace on a shoe, just like tying up the RBDs in a close conformation together, probably increasing the opening of the RBD that needs to be open in order to be functional. So increasing the energetics of the opening of RBD would actually make the RBD or the corresponding spike less infectious. And this is something that has been reported in a recent bioarchive, that a loss of N370 glycosylation leads to an increased replication in human cells. And also, curiously, the different strains that actually carry the N370 glycosylation are not, or have not been resolved by cryo-EM in an open form, but only in the closed form, such as the pangolin and the bat RATG13. So to finish it up, so this, I hope I convinced you that the spike-glycan shield is intrinsically involved in regulating the glycoprotein stability in its active form. So the type of glycosylation and the topology of the glycan shield affect the stability of the open conformation, the RBD opening energetics, thus as to binding and infectivity of the virus. So what is particularly interesting to us, and actually a line of work that we are pursuing at the moment in different directions, is that the cleft that is occupied by the N370 glycan on the closed RBD is now vacant, now in COV2 for the first time, is not occupied, and can be potentially occupied by other glycans. We've seen all evidence of GAGs binding the spike, the COV2, the spike, like sialylated glycolipids and different other glycans also might bind this cleft. So, and this will help cell surface localization for sure, as those glycans actually cover the cell surface. And this is a strategy that many viruses have used, so it's also accessible to COV2 now. So also, all this is a perspective because of the glycan shield identity or type and topology affects the stability of the open conformation. Also, targeting glycosylation looks like a very promising therapeutic strategy. And this was discussed by Spencer Williams and Ethan Goddard-Borger, like in a paper last year. So to conclude, like last but not least, I'd like to thank all my students that worked tirelessly through the year, doing really fantastic work, like for episode 1, episode 2, and episode 3 now, like so ongoing. So I'd like to also thank like my collaborators on episode 2, Ben Schultz and Toan Pung from University of Queensland, and from episode 1 and many other episodes to come, like our collaboration with Rami Amaro's group. In particular, I'd like to thank Rami, Lorenzo, Zayed, Abby, and Jason McLellan, who did the experiments for the Beyond Shielding paper. All other collaboration, and especially you for your kind attention. Thank you very much.
Thank you, Elisa, for a very nice talk. Excellent. Are there any questions for Elisa, please? You can just raise your hand if you have any questions, and we'll try to give you the word. No, so far. Okay, then I'll allow myself to have one ignorant question here. When you're doing this simulations and you're doing this quite large time scale, I mean, how long time does it take to compute, and how work intensive are they?
Very. So that's actually a very good question, because not a lot of people know that. Like I've been asked, is like, oh, can I do this on my laptop? No, like, so not a chance. So the latest simulations, so the ones that about the type of glycosylation, they, like for those, we have used one of the largest and most powerful supercomputers in Europe. So that is in Rome. So for that, we got a special allocation of 15.8 million core hours. And we ran for about 8 months constantly. And then what I would say, like, you know, like what is computer intensive? So you need a supercomputer to do this, and we run on GPUs. It's once they're done, you have terabytes of data that you actually don't know what you're looking at. So it's the analysis of data that takes quite a long time. So it took, overall, it took 10 months from the day we started to post the preprint.
And there's a question from Bernard. I think you could speak, Bernard.
And can you hear me?
Yeah.
Yes, we can hear you.
Hi.
Yeah, very good talk, Elisa. Really nice to hear you. I have a general glycobiology question here. but that can be applied to the glycan shield that you just talked about. In your opinion, is the role of the glycan shield something that has been evolved for by evolution, or is this just accidental?
Well, that's a very good question. So as the protein evolution, like it's, I think like for how I understand the problem, is accidental. So, and then whatever is more functional, or that it corresponds to the higher replication of the virus, wins. So it becomes the dominant strain. So because the glycan shield is functional and it responds to the stability of the protein and the characteristic of the protein, and it depends on the protein itself, actually it just undergoes the same rules. In particular, what we saw is that the N370 glycosylation was present for a while in the evolution of the this spike sequence. So, and meanwhile, the N234 glycosylation just came about recently, like in the like say phylogeny of the virus. So what we think, well, an explanation to this is just that 234, by filling that cleft, by being functional and propping up the spike, actually took the role of 370. Because, and that's more advantageous because 370 was great at propping up the RBD, but it was pretty rubbish when it was when the RBD needs to open, because it kind of laced it to the old kind of like closed envelope, or let's say like this kind of fold. So it took quite a lot of energy to get rid of that 1,6 arm. Actually, so much, and as you know very well, the binding affinity of glycans is like, let's say, notoriously low, like compared to other range of biomolecules. And during our many simulations, we never seen that 1,6 arm lifting up or changing conformation. So it's that corresponds to quite a bit of energy to pretty much unbinding and having the RBD open. So I think to answer your question in a very long-winded way, to summarize my answer, I'd say I don't have like, you know, a certainty of this, but I think in this case, the glycan shield has changed because it's functional, and it follows, those changes follow the same rules as the protein evolution of the virus. So whatever guarantees the highest reproduction of the virus then wins.
Yeah, so you mean it was not designed for, but it has been selected.
Yeah. Yeah.
Well, thanks a million.
Thank you. Thank you.
I'm going to let the other people ask their questions. Thank you so much.
Thank you very much.
Thanks, Bernard. I can see no other raised hands or anything like that. So I want to thank you again, Elisa. I really appreciate it. It was so nice to see someone in this field to teach us about this awful virus that we're battling right now. So it's very timely.